Hey everybody, welcome back. We're going to be talking about cell structure and parts of the cell. We're going to start with our prokaryotic organisms. Bacteria and archaea both have similar structure. So the most common types are coccus shaped and rod shaped, but there are multiple other shapes. We have Vibrio, which you can see right here, which is a curved rod. Spirillum, which are those spiral shaped. Spirochetes, which are a tight spiral. There are some cells that are called pleomorphic. So these would be pleomorphic cells where they have different shapes or they can change shapes. So you'll find different shaped cells in both archaea as well as in bacteria, but we're going to focus mainly on the bacteria. So bacterial cells replicate via binary fission, and when they replicate, oftentimes the cells, when they produce new cells, they stick together. So you have one single cell, it replicates, producing two cells, and when they stick together and they're coccus shaped, then they can form either diplococcus or streptococcus cells. And so each of these is an individual cell, but they are forming an arrangement, what we would call a multicellular arrangement, but they're still each individual cells. Sometimes the cells can form packets that look like, little cubicle or cubes. These are known as sarcina. Sometimes cells can form a square or diamond shaped structure. This strepto or your coccus cells can form this diamond shaped structure. That's known as a tetrad, which you don't see over here. And then we have the clusters that look like grape-like clusters, and that's known as staphylococcus. A lot of times the organisms are named for their shape. So like Streptococcus pyogenes is named for its shape. Staphylococcus aureus is named for its shape. I should mention, I'm showing here in coccus, but you can also have streptobacilli, which are chains of bacillus as well, which I didn't have a picture of here. So now let's look at what makes up a prokaryotic cell. So we have a cell wall, which is this blue structure. On the outside, we have most of our prokaryotic cells have some type of a capsule, not all, but many of them do. And then inside the cell wall, we have a plasma membrane. All cells have a plasma membrane. Not all cells have that cell wall. If we go internal the plasma membrane, we get the cytoplasm where we have the nucleoid and we have these little tiny structures known as ribosomes floating in the cytoplasm. Outside of the cell, prokaryotes can have these structures known as fimbriae or pili, and they can also, some have the flagella. So let's look at the plasma membrane. That's the first structure we like to look at. All cells have a plasma membrane, and the plasma membrane of every single cell is very similar. Plasma membranes are composed of phospholipids. So you can see a phospholipid here, which has a hydrophilic head. The term hydrophilic means that it is water-loving, water, philic, loving. And then it has two fatty acid tails. These are called hydrophobic tails. Hydrophobic means, again, water, hating. So they're scared of water. So we have two layers of these phospholipids. The hydrophilic heads face the external and internal environments which are fluid. The hydrophobic tails are sandwiched in between. Then we have proteins that are going to be found either on one side of the membrane or the other. Those are called peripheral proteins. and they have different functions. They might have functions to as enzymes. They might be regulating certain activities inside the cell. They might be identifiers. And then we have these other proteins that are spanning through the entire plasma membrane. Those are known as integral proteins. They are integrated in the plasma membrane. These are oftentimes channel proteins or transport proteins of some sort. So there are a lot of functions for proteins. I've named a few. They can be gates, they can be sensors. They work in the plasma membrane, though, to help in maintaining that membrane. The plasma membrane itself is considered a fluid mosaic. It is very fluid in its movement. The proteins drift about within the plasma membrane. The plasma membrane isn't very rigid or solid or hard. It moves with the environment that it's in. The plasma membrane has a very important function, though. It is the keeper of the cell. It protects the cell. It is a selectively permeable membrane that will allow small nonpolar molecules to pass freely through it. And then it allows other molecules to go through via aquaporins or through other active transport mechanisms. So through protein channels or other mechanisms, active transport or something like that. So I always liken the plasma membrane to the bouncer at a bar that allows certain individuals into the bar and keeps others out. And then if someone's getting too rowdy in the bar, it'll kick those people out actively, and maybe it keep someone who maybe was going to do something wrong in the bar against their will, again, actively, until the police show up. So that would be what the plasma membrane does. So here you can see simple diffusion. Diffusion is movement of molecules from one side of the membrane to the other. Molecules move back and forth via simple diffusion from higher to lower concentrations. Water is a special type of molecule. It is a hydrophilic molecule, but it can still move through the plasma membrane. The majority of water, though, moves through these protein channels called aquaporins, okay? Whereas large molecules and polar molecules, and so I'll use glucose as your example here, cannot pass through. Actually, that wasn't glucose, I guess. That might have been, I don't know, it was some type of a carbohydrate. So let's look at the different mechanisms for movement of materials across the membrane. The first is simple diffusion. Simple diffusion is movement of molecules from higher to lower concentration down a concentration gradient from higher to lower concentration. Simple diffusion is the movement of solutes, and the speed of movement is going to depend on the concentration. If you have a high concentration of molecules on one side of a membrane and a low concentration on the other, then then the solutes are going to move faster from higher to lower concentration. But if the concentration is much closer, then the molecules move slower. Osmosis is a form of diffusion. So osmosis is diffusion of water from a higher to lower concentration down a concentration gradient. But now we're moving water instead of the solute. So solute stays, water moves from higher to lower concentration. So now we're looking at concentration of water to solute ratio. If you have an environment that has a lot of water and few solutes, and an environment that has very little water and a lot of solutes, water moves from the hypotonic region into the region that is called hypertonic. until we have equilibrium of water on both sides. So it's not about having equal amounts of water, like you can have 100 milliliters of water on this side and this side. Water is going to keep moving until you have equal water to solute ratios on both sides. So in a hypotonic solution, the solution has lower solute concentration than the cell. So there's less solute out here, more solute in here. Water moves in, causing the cell to swell up. In a hypertonic solution, water, or there is a, the solution itself has a higher solute ratio than the cell has. So there's less solute in the solution or in the intracellular region, more solute outside. And so water leaves the cell until we have equal water to solute ratio on both sides. So, and then in an isotonic solution, we have equal amounts of solute and water on both sides. And so water is going to move in both directions equally. And you're not going to have a net gain of water on either side. In bacteria, because they have that cell wall, when the cell enters a hypertonic environment, water leaves the plasma membrane and leaves the cell. The cell itself, this plasma membrane, will pull away from the cell wall, and you'll see this plasmolysis form. Okay. If there's, and so that's what happens with the cell wall. If you have no cell wall, the cell will crenate. It just shrivels up and dehydrates. If you have no cell wall and you're in a hypotonic solution, fluids rush in at such a rate that the cell ends up bursting or lysosing open. Another form of passive transport is known as facilitative diffusion. Facilitative diffusion is a passive transport mechanism, but the molecules use some type of facilitator, so a protein channel that allows molecules to move through. And so facilitative diffusion is the movement of molecules from higher to lower energy, or not energy, I'm sorry, higher to lower concentration down a concentration gradient through a protein transporter or channel in the cell. We'll talk about these other mechanisms in just a minute, okay? Then we have active transport, and that's where these guys come in. Active transport is the use of energy to move molecules from one side of a membrane to the other. A lot of times this is against the concentration gradient. So movement of molecules from one side of a membrane to the other side of a membrane, utilizing energy against their concentration gradient, is active transport. So here you're seeing a secondary active transport where we have hydrogen ions at high concentration out here and low concentration down here. They're moving in. So this is using what we call a proton motive force as energy. So as the hydrogen ion moves across the membrane through that protein channel, another molecule moves across with it, uses the energy of this molecule to get across. So that's one mechanism of energy or of active transport. Another active transport mechanism is the ABC transporter, which utilizes ATP as the energy source. So in this case, ATP binds to the channel, and that allows the molecule to move from lower to higher concentration into or out of the cell, depending. So both of these are active transport mechanisms. And when we talk about this is a primary active transport. This is a secondary active transport. When I talk about proton motive force, we're talking about the electron transport chain. So electrons moving across a membrane from one side of a membrane to another side of a membrane, not electrons, hydrogen ions moving from one side of a membrane to another side of a membrane, utilizing the energy of electrons to allow them to pass back across. And so this is how the majority of ATP is produced in most eukaryotic cells and in many prokaryotic cells, including cells like E. coli and Staphylococcus. So this produces a lot of ATP energy, which is needed for multiple metabolic reactions, which we'll talk about more in a later chapter, okay? So this is the movement of those molecules down the membrane from higher to lower concentration, pulling other molecules. That's secondary active transport. But when we use the ATP from this electron transport chain, that's primary active transport. Oh, sorry. And the last one that we'll talk about is then group translocation. Group translocation is the movement of molecules from one side of a membrane to the other, but as they move the molecules, they alter the compound. So they change it. This is how they take in things like glucose. They pull the glucose in and they phosphorylate it at the same time. That is, that is what group translocation does, is it changes the shape of the molecule. All right, we're going to stop here. We'll get into the cell wall in our next video, okay? Bye.